Okay. Good morning, class. You should all be commended for resisting the temptation for a 4-day weekend. So, okay, so today we're going to move off what we were talking about yesterday in terms of the higher orders of protein structure. So today we're going to focus on the three-dimensional tertiary structure, and we're going to look at the types of interactions that are stabilizing that and the energetic. So hydrogen bonds, ion pairs, van der Waals, hydrophobic effect, and in some instances disulfide bonds. Then we'll kind of shift gears and talk about how it is that proteins are purified, analyzed, and characterized. All right, so we defined yesterday secondary structure as being the kind of localized configuration of amino acids that are close to one another in the primary structure. Tertiary is now the folding of these secondary elements into the three-dimensional structure. And in this case, we now have amino acids which may have been very distant in the primary structure that through the folding are being able to interact with one another. And in fact, a lot of those interactions can be stabilizing interactions, as we'll see. So this is what defines the complete 3D structure of a single polypeptide chain. The secondary structures, we said that they were stabilized with hydrogen bonds that involved main chain atoms. All right. For tertiary, we have a set of interactions, excuse me, that can stabilize it, and in some instances it is with backbone atoms, and in other instances with side chains. Okay. So remember that the side chains don't contribute to stabilizing secondary elements, but they do in the 3D structure. Okay, so just to kind of give you a little view of nature, we talked yesterday about alpha helices and beta sheets. So with the three secondary elements, alpha helices, beta sheets, beta turns, you might think, well, that's not much to work with in terms of making 3D structures and having a lot of variability or diversity. But in fact, you can mix and match those in various ways. So this is just to kind of give you an idea. You've got proteins which can be predominantly alpha helical. And in these cases, we have what's called helix bundles. So you have multiple helices coming together. If you did something like that, you could guesstimate as to what types of interactions might bring those helices together. You have proteins that are predominantly beta sheet. So in these cases, these are actually beta barrels. So we talked about yesterday how the multiple strands can come together and interact to form a sheet, right? But if you remember that figure that basically looked like a piece of paper with the R groups pointing up and down, you can have the beta strands moving around so that you actually have a barrel structure, which is what you have in these. They're anti-parallel. How do we know they're anti-parallel? When you see a beta, when you see a line like that with an arrowhead on it, is that a beta sheet or a beta strand? It's a beta strand. So if you're following the polypeptide chain, all that tells you, right, is that in that region, you've got a beta strand which has the configuration of the R groups going up, down, up, down, up, down. So then the question is, how are those beta strands coming together? They can come together in kind of a linear array to give you a sheet, but they can also come together like this, where they're kind of twisted and aligned and moving around. Now, in this case, these are examples of proteins which have a good mix of both helices and beta sheets. And the major thing is to see that, again, you can determine whether something is parallel or anti-parallel by the arrows, right? If the arrows are going in the same direction with the interacting beta strands, then that is parallel. Opposite, anti-parallel. Now here, if you look at this structure, you look at that, this is a twisted beta sheet. What you'll see is we focused on the beta sheet per se, which really was a sheet. But it's a little bit more complicated than that, because if the beta strands are interacting, right? If they are interacting with each other, the stabilization of those interactions are what? Hydrogen bonds between the strands. If they're not perfectly aligned to make a beta sheet, you can still have something like this, right? Would you call that a beta sheet? And in the future, I may kind of modify the notes a little bit. It is and it isn't. It's not a perfect sheet, bless you, but you have these beta strands that are interacting with each other. They're overlapping. They're forming a secondary structure. It's just not like a piece of paper, okay? So when you see beta strands interacting like that, we can call them a beta sheet as long as you appreciate the fact that they're not necessarily in all cases a perfect sheet, like a piece of paper, all right? And in this case, you see it's kind of twisted. The main point here is to realize that there's quite a bit of diversity in the way the beta sheets and the alpha helices can comprise the 3D structure of a protein. And also that when you look at a structure, you know that's an alpha helix and these are beta strands. And whether they be parallel or anti-parallel are dependent upon the arrowheads. All right. So what types of interactions will stabilize the three-dimensional structure? Hydrogen bonds, which you should already be familiar with. An ion pair or a salt bridge. All right, so an ion pair is where you have two full-blown opposite charges interacting. So for example, a lysine side chain that's positively charged, a glutamic acid side chain that's negatively charged. When those interact electrostatically, they're not slight electron deficiencies or excess as you would see in a hydrogen bond, but here they're full-blown charge differences. That is an ion pair or a salt bridge. Van der Waals interactions. I'll go over this in a moment. They're weak interactions. They're transient electrostatic interactions. And in many cases, they're between fixed or induced dipoles. So I'll give you an example of an induced dipole. The point here is they're very weak interactions. So for them to contribute significantly to stabilizing a 3D structure, you need a lot of them. Right? And we'll go over that. Hydrophobic effect, we've gone over this a number of times. We have the increased entropy of the solvent molecules. Bless you, Jesus, everybody has a cold today. Or allergies. And then in some cases, disulfide bonds. All right, so Van der Waals interactions, what are they? They're a weak force of attraction between electrically neutral molecules. And for our purposes, we're really interested in understanding them within the context of hydrophobic side chains interacting with each other. So if you have hydrophobic side chains interacting with each other, how can you envision that there would be any electrostatic interaction right? When we talk about electrostatic interactions and them being favorable, are we talking about enthalpy or entropy? Not the hydrophobic effect. If we, electrostatic, right, is enthalpy. Charge interactions that are favorable. So when you look at something like that, you should be able to say that is hydrophobic, right? Carbon and hydrogen. How can you get things interacting with each other that are electrically neutral, right, and have some favorable enthalpy? What do we know about electrons? They're moving, all right? You might know their momentum, you might know their position, but you're probably not going to know both. So as these electrons are moving in their shells with, let's say, two atoms that are approaching each other, let's say these are hydrogens, these are carbons. At any point in time, the electrons compared to each other and, let's say, the positive charge here for the proton, they're going to be in different positions, and at times their positions are going to be favorable. So, for example, here, if these two hydrogen atoms are interacting with each other, if the electrons at some particular point in time, a nanosecond, whatever it is, are in proximity, that's not favorable, right? Here, you're not really getting anything favorable. But imagine a scenario where in a split second time, and remember these interactions are transient, when you look at this, would you envision that there would be some favorable electrostatics in this interaction? Right? There is an asymmetry. They don't have full-blown charge, right? When you have something that is electronegative, let's say oxygen, and it's pulling the electron away, it's making that proton perpetually electron deficient. But when two protons come together, their electrons are spinning. Sometimes they're not going to be favorable. They're not going to like each other, but they can kind of get in sync sometimes, and this is what we mean by an induced dipole, where the electrons will be moving around and they'll kind of get in sync. And when they're in sync, you'll have these types of situations. So that would be, let's say, with the side chains of the aromatic amino acids. The aromatic amino acids, those ring structures, the electrons are moving around, right? If you get, let's say, two phenylalanines coming together and approaching each other in the interior of a protein, the electrons moving around can kind of induce dipoles in each other, and what's going to be favorable is that they're kind of in sync in a way that's going to make these types of interactions possible, right? So the important point here is to understand that these molecules are electrically neutral, and when we talk about van der Waals, we're talking, for our purposes, really focusing on the interactions of the hydrophobic side chains in the middle of a protein, or the hydrophobic side chains in the fatty acids that we'll talk about that make a membrane. They're weak interactions, right? But if you have a lot of them, then they're going to contribute favorable enthalpy, all right? And that attraction is very weak, but you need a lot of them. Is this clear or not? Okay? Okay, so hydrogen bonds, you should be pretty familiar with this. Here's an example of lysozyme with 3D structure being stabilized by hydrogen bonds. Really, the important point here is to realize that there are three flavors. In contrast to secondary elements where the hydrogen bonds are exclusively between the main chain atoms, now upon the folding, you can open it up where you have, let's say, side chain to side chain hydrogen bonds, okay? You can have side chain to backbone, and backbone to backbone. All right? So it's not limited just to the main chain atoms. Okay, so we talked about hydrophobic residues liking or driving the folding of a globular protein. We're going to get contributions by hydrophobic residues being buried, both favorable enthalpy and entropy. Okay? We talked about this, the increased solvent entropy. As you have those hydrophobic side chains, and they are being exposed to water, what are they doing to the water? They're stressing them out, they're limiting their ability to maximize their hydrogen bonds. So if the hydrophobic guys bury into the middle of a protein, then one of the contributions to stabilizing that folded structure is the increased solvent entropy of the water by sequestering those hydrophobic residues in the middle of the protein. Where do we get favorable enthalpy? Van der Waals. Okay? So we initially focused on the driving force for that self-association being increased solvent entropy, and that is true, but you have to appreciate the fact that when all those hydrophobics get together and they bury inside the middle of a protein or within a bilayer in a membrane, that you're also getting favorable enthalpy coming from those van der Waals interactions. All right, so if you take the primary structure, if you had a protein that was made from amino acid 1 to amino acid 300 and whatever, and you just had it strung out in kind of a random linear array, for that to fold up, is that going to be favorable energetically with regards to entropy? Just focusing on the protein. Is the protein more or less disordered in the folded state? Less disordered, more ordered. Is that favorable with regards to entropy? No. So when you look at the delta G for this adopting a conformation, delta S is favorable when it's positive, right? If you have a decrease in entropy, that is a negative term, right? And the delta G, remember, is delta H minus T delta S. So if the delta S is negative and it's being multiplied by T that's negative, then that's going to be positive. So this, when we look at the energetics, this is the delta G just for the conformational folding of the protein, looking at just that. So that term is saying, no, this is not a favorable thing to do. So what are the other parameters that are going to contribute favorable energetics to allow this to happen or not? The hydrophobic effect, right? That delta S here is the entropy experienced not by the protein but by the solvent. Why? Because it is a system. So this is going to contribute favorable delta G. The delta H for folding, if the delta G is negative, that's good. If the delta H is negative, that's good. And where is this delta H for folding coming from? Van der Waals, electrostatic interactions, hydrogen bonds, disulfides in, not usually. Usually the favorable enthalpy and entropy will bring it together, and in many cases the disulfide is for a protein that is extracellular or being secreted. And so that disulfide kind of, once it folds, it locks it into place, right? But it's good that you appreciate that the delta H is coming from many sources. And so when you add all those up, you get a net delta G that is negative, and that's why it happens, okay? So you should be very familiar with all these types of interactions and how they're contributing to favorable energetics to allow the proteins to fold. Okay. So this is an example, and this is not, it's not applicable to all proteins. The important point here, and I think I mentioned it yesterday, is that all of the information to make a protein is in the genetic sequence. So once those amino acids come together, I mean, in some instances, you have proteins that are going to come along and help it to fold. In many of those cases, they're kind of stabilizing intermediates. But for the most part, that linear structure, that linear sequence, those amino acids and that backbone is going to know, for lack of a better term, how to fold up. It could try multiple ways, but the three-dimensional structure that it finally adopts is really the most energetically favorable product. So things are going to funnel down in the direction of what is the most favorable conformation energetically. So this figure is simply just meant to show that in a lot of cases, like RNase A, you can take the protein and denature it. You can heat it up, and it's going to unfold. And then as you slowly bring the temperature back down, it will refold and regain activity. All right. There are many proteins that don't do that because they're put in a slightly different situation than when they're made in the cell. When they're made in the cell, and we'll go over this, but you should have some understanding of it already. The protein is being made on the ribosome, so it's coming out and amino acids are being added one at a time, right? So that means that a portion of the protein would have been synthesized before the rest of it. So you could get some types of interactions there that temporally, right, would happen before. In other words, you could have amino acids that are at the beginning of the protein, and they don't really have the opportunity to interact during the folding process with amino acids at the end of the protein because they haven't been synthesized yet. But in some cases, if you take a protein that's a full structure, you denature it, that's kind of an artificial situation because in the cell, you don't have that situation where the entire protein has been synthesized and now you're asking it to fold. In the cell, the protein starts to fold as it's being made, okay? But the important point there is to think that, to understand in many cases, you can denature a protein and it will fold back, and regardless, the information for how it's going to fold is there in the primary structure. All right, protein misfolding. It's not a good thing. We will see in sickle cell how we have abnormal folding of a protein, but there are a lot of diseases that have either been shown or the evidence is pretty strong that the basis for the disease is protein misfolding, all right? And you can obviously read more about those. The important point is to realize that, you know, folding plays an important role in the normal function, the normal structure, which dictates function of enzymes, but also misfolding can create things which are toxic and bad. All right, so now we're going to switch gears and talk a little bit about protein purifications and how we kind of deal with proteins. This is a classic quote from Arthur Kornberg, a Nobel Prize winner: "Don't waste clean thoughts on dirty enzymes." Essentially, in the early days of biochemistry, you're trying to figure out how things are acting, what the reactions are like, characterizing life in a test tube, and if you're not working with stuff that's clean, then you shouldn't waste your time working on it. So when we're going to purify a protein and characterize it, first of all, the 3D structure is going to determine or dictate the properties of the protein. So as proteins vary in their DNA sequence, they're going to vary in their primary structure, which is going to dictate differences in three-dimensional structure. So they're going to be different sizes, different conformations. They're going to have different charges based on what the composition of the proteins are, where the charges are located. Are they predominantly basic? Are they predominantly acidic? So the proteins are purified. Once you have a purified protein, you can get the amino acid sequence for at least a portion of it. And once you have that, with knowledge of the genetic code, you can go back and look for the gene. So we'll talk a little bit more about this process of reverse genetics, right? In the old days, real old days, before biochemistry really was moving forward, you would identify a gene and you would say, I have this gene that I've identified genetically, right? The genetics would identify mutations in genes that were doing this or that or having changes in the way a cell behaves. So you would then try to understand what was coming from that gene, right? In reverse genetics, you're basically starting with a protein, and you're purifying the protein, saying, we're interested in this function, this activity, and now that we have it, we're going to extract some information from the protein sequence, and then identify the gene for it, right? So we'll talk more about that on, did I say Monday? I meant Tuesday. Okay, and so if you want, to know a little bit more about this, look at the tools of biochemistry at the end of chapter 5. All right. So we're going to briefly go over some of the strategies and approaches during protein purification, and then a determination of its primary structure. And we don't need to know, and we never really use these biochemical approaches to determine the entire primary structure of a protein. Just a portion is what we need to be able to do the reverse genetics. So we'll talk a little bit about purification, how we break it up, and do some sequencing, which is the Edmond procedure, and then with that information, we can try to identify the gene. All right, for all of you potential blossoming biochemists out there, if you're starting with, let's say, an activity, let's say you identify some activity in an extract and you want to purify it, you don't know what protein it is, right? You don't know what gene it's coming from. With the human genome being known, you know what's out there, but you don't know if you start out with something functional, something biochemical, who it is you're dealing with. So if you're going to purify it, the first thing you have to think about is, where am I going to get my protein? What's the source, the cells, etc. As you're purifying a protein, what is the assay for it? So in some cases, we can purify a protein where we're not doing, let's say, reverse genetics. Let's say we know what gene it's coming from, but we want to make a lot of that protein. And maybe that protein has to be purified from its source, right? We can't do it in a recombinant way where we put it in a clone, put it in bacteria, tell the bacteria to make a lot of it. The point is, as you purify something, you have to follow it, right? You have to follow the protein, and then you also have to look at the other proteins that are coming along with it. In theory, if you're purifying it, you're getting your protein from step to step to step, and what's happening with the other proteins. They should be going away. So when we talk about specific activity, that is the activity or the amount of our functional protein with respect to the total amount of protein that's there. So we want something that has high specific activity. We want something that is greatly enriched for our protein of interest. So during the purification, you have to be able to follow your protein of interest. If you already know who it is and you have antibody, you can do Western blotting, for those of you who know it, but in many cases you have to just do a functional assay. Whatever that enzyme does, you have to say how much activity do I have, and then how many proteins are there. Okay, so principles of column chromatography. Let's say you have a mixture of proteins and you want to start fractionating them. You want to purify your protein of interest away from the other ones. The basics of column chromatography, and as it implies that there's a column, which there's in the next figure, it's depicted. You have two phases. You have something which is in that column, in that tube that's packed, which we'll call a resin, or we'll call the stationary phase. The protein initially is in the mobile phase or the liquid phase. So that might be a lysate, it's in a buffer, and you're going to apply that to the column that is packed with the solid phase or the resin. You're then going to start passing buffer through, and all of the proteins are going to be moving through that column. So how they interact with that column and how they're separated, that can be done using really one of three different approaches, right? But the idea is you're getting fractionation, right? You're getting fractionation of proteins. Some are going to interact with that stationary phase well, and they're going to get hung up on the column. So as the mobile phase is going through, they're going to spend more time interacting with the column, and they're going to come out the bottom of the column later, right? So this material that's in the column, we'll call the resin or the stationary phase, that initially you might have this mixture of proteins, you're putting it on top of the column, and you're running a buffer through. You're getting fractionation. These proteins are interacting with that solid phase to different extents. Those that aren't interacting strong are going to come off of the column early, right? So we call this fractionation, and these individual small volumes that are taken as a function of time are fractions. And then those that have a stronger interaction with the column are going to come off later. And in many cases, well, not in many cases, in two of the three cases, the buffer that we're running through the column, we will change the composition of to start to knock proteins off, and you'll see that in a minute. All right, so the first type of chromatography is ion exchange. So the stationary phase in ion exchange is charged. It has a permanent charge on it. So in the manual, there's, I think I put it in the notes, a couple of structures there. It's also in the text. But the basic idea is that the resin that is in there, these beads are coated with something, bless you, which is either very positively charged or very negatively charged. All right? So if the beads are positively charged, right, what types of proteins or what types of charges are going to bind to that resin? Negative, right? An anion. So things that are positively charged, we say are anion exchange columns, because the resin itself is positive. Things that are highly anionic, negatively charged, are going to bind, right? And we say exchange because in this type of chromatography, if you have a highly positively charged resin, and then you put an extract over and you have a protein that's very negatively charged, as it starts to go through the resin, what's it going to do? It's going to stick. And if the interaction is tight, which in these types of chromatography they are, they're going to stay on there. If the buffer that you put on, it binds, if you keep passing that same buffer through, those proteins that stuck remain stuck. So how do we get them off? change the composition of the buffer, and we'll talk about that in a second. So, we'll talk about it here. In ion exchange, you can have columns that are positively charged, which are going to bind negatively charged proteins, or they're negatively charged, which are going to bind positively charged proteins. Now, when we say positively charged, negatively charged, what is it that we're really talking about? No, with the proteins, the R groups, all right? So highly charged R groups are not going to be found buried away in the interior. They're on the surface. So if you have a protein that has, let's say, a lot of negative charge on the surface, a lot of glutamics, a lot of aspartics, then they're going to interact with this column and they're going to bind. Let's say you have another protein. Let's back up. Let's say we break all the proteins in this lysate in kind of two flavors. Those that are pretty positively charged, those are pretty negatively charged. Within each one, you've got some that are very, very highly charged, intermediate, low intermediate, low, ultra low, whatever. It's a spectrum. So let's say we have an anion exchange column. We pass the lysate through. First of all, who do we know is going to fly through the column and basically come out when the buffer comes? Huh? Positively charged proteins. The positive charged proteins are not going to interact with that resin, so they're going to come flying through. So now everybody else who's there, and in this hopefully we're saying our protein is one of them, is there on the column. If we keep running the same buffer through, for the most part, they're all going to stick there. So now what we do is we take a gradient of increasing salt. So salt is used to elute proteins off of ion exchange. All right? Why do we call it ion exchange? Typically, this salt that we'll use will be, let's say, potassium chloride. Potassium, positive ion, chloride, negative. So as the KCl comes through, we run a gradient so that the buffer that we're applying to the top of the column, we slowly raise the concentration of the salt. So as we start to slowly raise it, all right, we've got a positively charged resin. That means proteins that have negative charge on their surface are bound to that, but they're going to be bound to various extents depending upon how much negative charge. So let's say we have some proteins that have a little bit of negative charge. They're going to be the ones that are going to be most easily displaced off the column, right? And what is it that's going to exchange for the protein on the resin? The negatively charged chloride. So that's why it's called ion exchange. The ions are either from the side chains that initially bind it, and then at the time of elution, the ions are coming from the salt. Is that clear? So then, when you do this gradient, proteins that bound but have a kind of a low amount of negative charge, they're going to start coming off the column early, let's say at 125 millimolar salt. Then as we raise the salt in a linear fashion in a gradient, proteins that have had slightly greater amount of negative charge, then they come off. And the ones that come off last are going to be those that had what? The highest amount of negative charge. So in ion exchange, you're separating charges, you're separating the proteins based on their charge. And you've got two ways to approach it, with either a cation exchanger, which is negatively charged on the resin, right, or an anion exchanger, which is positively charged on the resin. Okay, gel filtration chromatography. This is based on separating them by size. So in this case, we do not change the composition of the buffer. In the column, we have these beads that have defined pore sizes. And when we apply a mixture to the top of the column, if we look over here, how many of you golf? Only one person? All right. How many of you have practiced with a plastic practice golf ball? Like the tiny little wiffle ball? All right. So this column is filled with little tiny beads that kind of look like that. And the hole size on the ball can be different, right? You can buy gel filtration resins where the hole size on the beads can be different, and depending upon who you're trying to purify and so forth, you can buy different types. But the idea is this. Let's say this is your practice golf ball and you have all these little holes. If I present a mixture of proteins to those packed beads in the column, if a protein is small, it's going to be able to get in that little golf ball, right? And it's going to bang around in there for a while. As you're passing the buffer, it's going to bang around in there for a while before it finds its way out. But then it's going to find another one, and it's going to go in there and bang around for a while before it can get out. So as you're passing the buffer through, proteins that are small are going to spend a lot of time inside those beads. They bang around for a while, they come out, they get another one, they get hung up, right? What about the bigger proteins? If a protein is bigger than the pore size, it's never going to get in, and it comes out what's called the flow-through, right? Whatever the volume of liquid is in the column, if it never enters the bead, then as you're passing more and more buffer, it just kind of bangs around the outside of them, never spends any time inside the bead, so it comes off when that volume of buffer comes off. Within those two extremes, something very, very small and something that never spends any time in the bead, for those that can enter into the bead, the amount of time that they spend caught up in one bead before they find their way out is going to be different from protein to protein, okay? And so in that sense, you're going to get a separation based on size. Proteins that are very big, that never go in at all, right, are going to come out first. Then those that are big but got in are going to come out next. Then those that are medium big are going to come out next. Those that are medium that are going to come out next, down to the smallest ones, okay? So ion exchange based on charge, gel filtration based on size, affinity chromatography based on really kind of function. So in affinity chromatography, what's on the stationary phase is something that is very specific for what that protein likes. Right? So if it's an enzyme, sometimes you could actually put the substrate immobilized on there. And when you pass this complex mixture of proteins on, your desired protein is going to bind this affinity tag. So in the case, let's say, of an enzyme, it could very well be its substrate. Today, what we do when we're doing production of proteins in large amounts where we have the gene cloned is we can clone the gene, and then at the beginning, at the end terminus, we can put a little tag. So something that's used quite a bit, it's called a histidine tag, 6 histidines. You don't have to know this, but the idea there is we can then give that to bacteria, turn the protein production on, make lots of it, break the cells open, and put it over a column that has immobilized nickel or immobilized cobalt, which will bind to those consecutive histidines. And so in that case, we're introducing some type of a tag on the protein that binds something that's on the resin, right? But the important thing for affinity chromatography to know is that in contrast to separating them on size or just general charge, you're doing it based on something specific on the protein that is going to bind specifically to something on the resin. Okay, so those are some of the approaches in terms of purifying. But through the whole thing, we want to follow our protein. We want to see what the protein mixture is. let's say you were doing a purification and you were doing it based on the activity of an enzyme. As you go through these different steps, when you're coming off these columns and you have all these fractions, where do you know your protein is? Let's say you don't have an antibody or anything. If you're purifying an enzyme that has a particular activity, it carries out a certain reaction, then in these columns, you would start with a lysate and you say, I know my activity is there. So now I'm going to put it on a column, I'm going to do my fractionation, let's say a cation exchanger or anion. You would then look at maybe every other fraction and do that assay and say, where is my activity? It's here. So now I'm going to take this and now put that on another column, which is going to separate those guys based on something different. So in a purification, you might have one type of ion exchange, followed by something different, followed by gel filtration, etc. But the point is, if you get your activity and you're recovering it, but you're not getting rid of a lot of proteins, you're not really purifying it that much. So you want to follow your activity, but you want to follow how complex the protein mixture is at every step in the purification. So SDS protein gels, how many have heard of these? You should have. Maybe not. So real quick, in electrophoresis, where you have some type of matrix that is a gel, sometimes it's between two glass plates, as it is in SDS Page, normally, if you would take biomolecules and just put them on some type of a gel, which is a matrix, which is basically cross-linked and has little pores, so things are going to kind of move through. If you apply an electric field, how they go through that matrix is going to be dependent upon how big they are, right? How much charge they have and what their shape is. Something that's kind of tight and in a tuck position is going to be able to go through those pores a lot better than something that's kind of floppy and getting its arms hung up. So in SDS Page, what you're doing is normal electrophoresis of just straight native biomolecules through a matrix. Those three things are going to determine how quickly they go through. But in SDS Page, when you have a protein, SDS is a detergent, right? It's got a hydrophobic tail. It has a hydrophilic end, so that is what type of molecule? Amphipathic. You heat it up, the protein opens up like this, the hydrophobic parts extend towards the backbone, you have a lot of negative charge, all right? The mercaptoethanol in there is to break disulfides, heat to denature it. But the point with SDS Page then is you give all the proteins essentially the same shape, rod-like. You give them such a high amount of negative charge by the amount of SDS that binds that for all intents and purposes, the system doesn't recognize the differences in the charges. So that the final parameter that they're separated on is size. So in SDS Page, as you run these through, the large proteins will run higher up in the gel, the smaller proteins will move faster through the gel. A couple of minutes. 2D gels, won't go over this too much other than to say that if you run proteins on a 1D gel, there are a lot of proteins that may be the same size and they'll kind of overlap each other as a band on a gel, but really that band is many protein species. So another parameter, okay, is isoelectric point. So isoelectric point is if you look at a protein, it's going to have a certain amount of charge on it, right? It could be basic, could be acidic, more neutral. What kind of charge it is going to, the amount of charge that it has on it is dependent on two things. Its composition and what? pH. All right? So when you do these fractionations and so forth, the buffer pH that you choose is important. But the important point here for isoelectric focusing is that there's a pH at which every protein has an overall net zero charge, where all of the acidic side chains that are dissociated are balanced by all of the positive basic side chains that are positively charged, right? If you have something that is if you have a general protein and the pH is very, very high, you're going to get a lot of dissociation, so it's going to be more on the negative side. If you lower that in pH, things are going to start protonating, and so they're going to become either neutral or positive. So somewhere in between there is a pH at which all of the positive charges in the protein balance all the negatives, and then its overall net charge is 0. So that, by definition, is the isoelectric point, the pH at which there's no net charge. You can look in the notes here, but basically the way this is done is you separate them first by isoelectric focusing, and then you take that gel, stack it on a normal SDS gel, and separate it by size, okay? All right, so on Tuesday, we'll pick up with the Edmond degradation and how we go into the reverse genetics. Have a good weekend.